Today's topic is antimicrobial susceptibility testing. Before that, we will see about antimicrobial agents and antimicrobial resistance. Learning objectives are the classification of antimicrobial agents, intrinsic and acquired antimicrobial resistance, methods of antimicrobial susceptibility testing. Agents that kill or inhibit the growth of microorganisms that are called as the antimicrobial agents. Now, classification of antimicrobial agents. There are 4 classifications. First one, according to the microorganism against which they are used. Microorganisms like bacteria, they are called antibacterial. Fungal, that they are called antifungal. Parasitic, that are called antiparasitic. And against viral, the agents are called antiviral agents. According to their ability to kill, second classification is there, or inhibit the microorganism, that is a bactericidal and bacteriostatic. Third one is according to the source of antimicrobial agents, that are the antibiotics and chemotherapeutic agents. Fourth one is according to their site of action and uses. They are the disinfectant, antiseptic or antibiotics. Now, mechanism of actions of antimicrobial agents. They act on cell wall or peptidoglycan synthesis. They are the bacitrazine, glycopeptides, phosphomycins, vancomycin and beta-lactams. They act on cell wall or cell wall polypeptidoglycans. Second one is a protein synthesis. Proteins are 30S and 50S. Protein synthesis inhibitors. First one is 30S inhibitors, that are the aminoglycosides, tetracycline and tricycline. 50S inhibitors are chloramphenicol, clindamycin, linezolid, macrolides and streptogarnins. Cytoplasmic membrane inhibition, they are the polymyxin B and daptomycins. Nucleic acid synthesis inhibition, they are the fluoroquinolones by decreased DNA gyrase, metronidazole, they also decrease DNA synthesis, and rifamycin, they decrease RNA polymerase. Inhibit metabolic pathway of folate or folic acid. They are the sulfonamides, trimethoprime and cotrimazole. Now, classifications, indication and mechanism of actions. First class is penicillins. Drugs are penicillin G, aqueous penicillin G, procaine penicillin G, benzathine penicillin G and penicillin V. Spectrum of activity is they inhibit the cell wall synthesis. Gram-positive bacteria, that are the Streptococcus pyogenes, Pneumococcus, Corynebacteria diphtheria, Clostridium tetani and Clostridium perfringens. Others are meningococcal infection, gonococcus or Trypanosoma pallidum against the syphilis. Mechanism of resistance is drug inactivation by producing beta-lactamase enzyme, alteration of target site, and third one is a decreased permeability. Second class is penicillinase resistant penicillins. Drugs are methicillin, oxacillin, cloxacillin and dicloxacillin. Spectrum of activity, they inhibit cell wall synthesis same as penicillin plus penicillinase producing Staphylococcus aureus. Mechanism of action is drug inactivation by producing beta-lactamase enzyme, alteration of target site and decreased permeability. Aminopenicillins, extended spectrum, they are the ampicillin and amoxicillins. Spectrum of activity is same as penicillin plus Enterococcus faecalis, Escherichia coli, Klebsiella, H. pylori, Salmonella and Shigella. Mechanism of action is same as penicillin resistant penicillins. Anti-pseudomonal penicillins. Drugs are carbanicillin, ticarcillin and piperacillins. Inhibition of cell wall synthesis same as aminopenicillin is. Mechanism of action is same as the drug inactivation by producing beta-lactamase, alteration of target size and decreased permeability. Class beta-lactam plus beta-lactamase inhibitors. Drugs are aminopenicillins, anti-pseudomonal penicillins plus clavulanic acid, salbactam or tazobactam or avibactam. Spectrum of activity same as aminopenicillins or anti-pseudomonal penicillin spectrum plus beta-lactamase producing bacteria. Mechanism is same. Now, cephalosporins, they are the first generation, second generation, third generation, fourth generation and fifth generation. Inhibit cell wall synthesis is the spectrum of activity. First generation, cefazolin, cefalhexin. Spectrum of activity against Staphylococcus aureus, Staphylococcus epidermidis, some gram-negatives like Escherichia coli and Klebsiella. Mechanism of resistance is ESBL, that is extended spectrum beta-lactamase. In second, third and fourth and fifth generation, mechanism of resistance is same as ESBL. Second generation, that is a cefoxitin, cefaclor and a cefuroxime. Spectrum of activity is same as first generation plus increased gram-negative activity. In third generation, drugs are ceftriaxone, cefotaxime and ceftazidime. Spectrum of activity, that is a decreased activity against gram-positive compared to first and second generation. Increased gram-negative activity, some are active against pseudomonas, that are the ceftazidime. In fourth generation, drugs are cefepime and cefpirone. Good activity against gram-positive and gram-negative, including pseudomonas. Fifth generation contains drugs are ceftobiprole and ceftaroline. Same as fourth generation plus pseudomonas and MRSA. Only beta-lactam to be effective against methicillin-resistant Staphylococcus aureus. Carbapenems, drugs are imipenem, meropenem, doripenem and ertapenem. Spectrum of activity is inhibit cell wall synthesis, that is the broadest range of activity against most bacteria which include gram-positive cocci, Enterobacteriaceae, Pseudomonas, Listeria, anaerobes like Bacteria fragilis, Clostridium difficultis, except MRSA and Mycoplasma, etc. Mechanism of resistance is that produces carbapenemase and efflux pump. Aztreonam, drug is aztreonam. Spectrum of activity is gram-negative rods, aerobes and hospital-acquired infections. Mechanism of resistance is extended spectrum beta-lactamase. Now, glycopeptides, that is bactericidal, disrupt peptidoglycan cross linkage. Drug is vancomycin and teicoplanin. Other cell wall inhibitors that active against most gram-positive MRSA and indicated if the patient is allergic to beta-lactam drugs. Mechanism of resistance is alteration of target site. Phosphomycin. Drug is also phosphomycin. that inactivates the enzyme UDP N-acetyl glucosamine 3-enolopyruvate transferase, also known as a MURA, required for cell wall synthesis, active against urinary tract pathogens, against both gram-positive and negative bacteria such as E. faecalis and E. coli. Mechanism of resistance is alteration of target sites producing enzyme that inactivate phosphomycins. Other class, that is a bacitracin drug, is also bacitracin. Spectrum of activity is tropical gram-positive cocci infections. Mechanism is not defined. Now, aminoglycosides, bactericidal, irreversible binding to the 30S ribosome. Drugs are gentamicin, neomycin, amikacin, tobramycin and streptomycin. Anti-30S ribosomal subunits spectrum of activity is aerobic gram-negative bacteria often used for empirical therapy in an adjunct with third-generation cephalosporin in respiratory tract infections, meningitis and subacute bacterial endocarditis. Mechanism of action resistance is drug inactivation by aminoglycosides modifying enzyme, decreased permeability through gram-negative outer membrane and decreased influx of drug. Now tetracyclines, that are the bacteriostatic, tetracycline, doxycycline, minocycline and democlocycline. Protein synthesis inhibitors is Rickettsia, Chlamydia, Mycoplasma, Spirochetes, Yersinia pestis, Brucella, Haemophilus ducreyi, Campylobacter and Vibrio cholerae. Mechanism of resistance is decreased intracellular drug accumulations, ribosomal target site alteration. Now class chloramphenicol, drug is also chloramphenicol. Spectrum of activity against H. influenza, pyogenic meningitis, brain abscess, anaerobic infections, enteric fever, not used now due to development of resistance. Drug inactivation by producing chloramphenicol and acetyl transferase enzyme, altered membrane transport through active efflux. Macrolides class, drug are erythromycin, azithromycin and clarithromycin against Streptococcus, H. influenza and Mycoplasma pneumoniae. Resistance is alteration of ribosomal target and active efflux of antibiotics. Lincosamides drugs are clindamycin, bacteroides fragilis, Staphylococcus aureus, excellent bond penetration. Resistance is by alteration of target site, ribosomal methylation. Linezolid drug is also linezolid, resistance against gram-positives like MRSA. Mechanism of resistance is alteration of target sites. Streptogarmin drugs are quinpristrine and dalfoprostine. Activity against Streptococcus pyogenes, Staphylococcus aureus, skin infections, MRSA infection and VRE infection, that is a vancomycin resistant enterococci. Resistance is alteration of target site, dalfoprostine, active efflux, drug inactivation in quinpristrine and dalfoprostine. Mupirocin, spectrum of activity against it is used as a topical ointment against skin infection and nasal carrier of MRSA. Mechanism of action is mutation of gene for target site protein. Now, nucleic acid synthesis inhibitor, they are the DNA synthesis inhibitors. Fluoroquinolones inhibit DNA gyrase A subunit and topoisomerase IV, thus inhibiting DNA synthesis. They are the nalidixic acid, coliform gram-negative bacilli. Fluoroquinolones first generation, they are the norfloxacin, ciprofloxacin and ofloxacin. Second generation fluoroquinolones are levofloxacin, lomifloxacin, sparfloxacin and moxifloxacin. Spectrum of activity against enterobacteriaceae such as E. coli, Klebsiella, Enterobacter, Salmonella, Shigella, Proteus. Others are Neisseria, Haemophilus, Campylobacter, Vibrio, Pseudomonas, Staphylococcus aureus. Mechanism of resistance is alteration of target sites, mutation of DNA gyrase genes. Second is poor transport across cell membrane. Now class metronidazole intracellularly generates metabolic byproducts that damage DNA. Spectrum of activity against anaerobic organism. Mechanism of resistance is not defined. Now rifampin inhibit RNA polymerase, RNA synthesis inhibitors like Staphylococcus and Mycobacterium tuberculosis. Mechanism of action of resistant is alteration of target site by mutation of RepoBG. Isoniazide drugs inhibits mycolic acid synthesis. Spectrum of activity is tuberculosis and latent TB. Resistance by mutation in enzyme processing isoniazide into active metabolites. Folic acid synthesis inhibitor, they are the bacteriostatic competitively inhibit enzyme involved in two steps of folic acid biosynthesis, PABA, para-aminobenzoic acid, folate synthesis, dihydrofolic acid sulfonamide blocks, and dihydrofolate reductase that cause tetrahydrofolic acid synthesis, trimethoprime blocks. Sulfonamides and trimethoprime drugs are sulfonamide, sulfacetamide and cotrimazole, trimethoprime plus sulfamethoxazole. Sulfadiazines used topically in burn wound surface. Cotrimazole is indicated in urinary tract and respiratory tract infection, active against Serratia, Klebsiella, Enterobacter, Shigella dysentery, Vibrio cholerae, Haemophilus ducrei, Pneumocystis cerevisiae and Toxoplasma gondii. Production of insensitive targets that bypass metabolic blocks. That is the mechanism of resistance. Now, gramicidin forms spore. Topical use against cocci, gram positive and negative. Mechanism of resistance is not defined. Daptomycin form channels that disrupt membrane potential. Spectrum of activities bacteriocellular against gram positive including VRE and MRSA. Mechanism of resistance is not defined. Polymyxin B is topical agent, colistin or polymyxin E. Gram negative infections and resistance of mechanism of resistance is alteration of lipopolysaccharides and efflux pump mediated. Now antimicrobial resistance refer to development of resistance to an antimicrobial drugs by a microorganism that can be defined two types, intrinsic resistant and acquired resistant. Intrinsic resistance, it refers to innate ability of bacterium to resist class of microbial agents due to its inherent structure or functional characteristics. For examples, anaerobic bacteria, aminoglycoside is the intrinsic resistant. Organism aerobic bacteria, intrinsic resistant against metronidazole. Gram-negative bacteria, vancomycin, Klebsiella species, ampicillin. Pseudomonas for sulfonamide, trimethoprime, tetracyclines and chloramphenicol. Enterococci, aminoglycoside and all cephalosporins. Serratia, Proteus and Burkholderia, polymyxin B and colistin. Strenotophomonas maltophilia, that is against carbapenems. Acquired resistance, emergence of resistance in bacteria that are ordinarily susceptible by acquiring the gene coding for resistant. Overuse and misuse of this agent is the single most important cause of development of acquired resistance. Use of the particular antibiotic possess selective pressure in a population which in turn promotes resistance bacteria to thrive and susceptible bacteria to die off. Resistant bacterial population flourish in area of high antimicrobial use where they enjoy selective advantage over susceptible population. Now we will see the factors favoring the spread of antimicrobial resistance, that includes poor infection control practice in hospitals. Like poor hand hygiene practice can facilitate transmission of resistant strain, inadequate sanitary conditions, inappropriate food handling, irrational use of antibiotics by doctors not following antimicrobial susceptibility reports, uncontrolled sale of antibiotics over the counter without prescription. Now, mutational and transferable drug resistance. In presence of selective antibiotic pressure, bacteria acquire new genes mainly by mutation of resistant genes, by transferable drug resistance by acquisition of new genes, by horizontal gene transfer mechanism like conjugation, transduction and transformation. Resistance developed due to mutation of resident genes, that is a mutational resistance. Mycobacterium tuberculosis against ATT. Low level resistance developed to one drug at a time, overcome by using combination of different class of drugs. Now, transferable drug resistance. It is a plasmid coded transferred by conjugation or rarely transduction or transformation. Resistance coded plasmid, that is a R plasmid, can carry multiple genes, each coding for resistance to one class of antibiotic, result in high degree of resistance to multiple drugs, which cannot be overcome by using a combination of drugs. Now, mutational versus transferable drug resistance. Resistance to one drug at a time is mutational drug resistance. Transferable drug resistance is multiple drug resistance at the same time. Lower degree resistance in mutational drug resistance, high degree resistance in transferable drug resistance. Resistance can be overcome by combination of drugs in mutational type. In transferable type cannot be overcome by drug combination. Virulence of resistance mutants may be lowered in mutational, virulence not be decreased in transferable. Resistance is not transferable to other organism but spread to offspring by vertical spread only in mutational drug resistance. In transferable, resistance is transferred to the organism spread by horizontal spread like conjugation, transduction, transformation. Now, mechanism of antimicrobial resistance by decreased permeability across the cell wall. First one, that is, a bacteria modified their cell membrane porine channels either in the frequency or size or selectively. That prevents antimicrobials from entering into the cell. Observed in many gram-negative bacteria like Pseudomonas, Enterobacters and Klebsiella species against drugs such as imipenem, aminoglycoside and quinolones. Second is efflux pumps mechanism mediates explosion of the drugs from the cell soon after their entry, thereby decreasing the intracellular accumulation of drugs, and observed in Escherichia coli, Enterobacteriaceae against tetracycline chloramphenicol, Staphylococcus against macrolide streptogarmins, and Staphylococcus aureus and Streptococcus pneumoniae against fluoroquinolones. Third one is by enzymatic inactivation. It inactivates the microbial agents by producing various enzymes such as beta-lactamase enzyme production that breaks down the beta-lactam rings, thereby inactivating the beta-lactam antibiotics. Aminoglycoside modifying enzyme like acetyl transferase, adenyl transferase, phosphotransferase produced by both gram-negative and positive bacteria. Chloramphenicol acetyl transferase. Fourth one, by modification of the target site within the bacteria. Observed in MRSA, target site of penicillins. Penicillin binding protein gets altered to the PABA2, PBP2A. Streptomycin resistance in Mycobacterium tuberculosis due to modification of ribosomal proteins or 16S RNA. In rifampicin resistance in Mycobacterium tuberculosis due to mutation in RNA polymerase. Phenolon resistance seen in many gram-positive bacteria, particularly Asorius pneumoniae due to mutation in DNA gyrase enzyme. Beta-lactamase enzyme hydrolyze the beta-lactam ring of the lactam antibiotics, thereby deactivating their antibacterial properties. Produced by both gram-positive negative organisms, plasmid-coded and transferred from one bacterium to another, mostly by conjugation. Beta-lactamase classification, Ambler's classification, Booth-Jacobi, Medier's classification or functional classification, that is phenotypic. First one, that is Ambler's. According to this, beta-lactams are classified into 4 classes. And Booth-Jacobi, this is the most advanced and complex classification. We will see Ambler's classification of beta-lactamase. Class A, that is extended spectrum beta-lactamase. Organism producing ESBL enzyme are resistant to all penicillins, first, second, third generation cephalosporin and monobactam. However, remain sensitive to carbapenem and cefamycin. Resistance can be overcome by use of beta-lactam along with beta-lactamase inhibitors, that is a sulbactam and clavulanic acid. Detected by combination tests like ceftazidime and ceftazidime plus clavulanic acid, 3-dimensional test is best method. Class B, that is MBL metallobacta-lactamase. This mechanism are resistant to all those antibiotics to which MPC beta-lactamase producers are resistant, plus they are resistant to carbapenems. That is, the resistance cannot be overcome by beta-lactam plus beta-lactamase combinations detected by EDTA dyssynergy test or modified Hodge test. Class C, that is the MPC beta-lactamase. These organisms are resistant to all those antibiotics to which ESBL producer are resistant, plus they are resistant to cefamycins, but they are sensitive to carbapenem. That is, resistance cannot be overcome by beta-lactam plus beta-lactamase combination detected by MPC disc test using cefoxitin disc. Class D, that is the oxacillin. Resistance can be overcome by beta-lactam plus beta-lactamase inhibitor combination. Now, antimicrobial susceptibility testing. Bacteria exhibit great strain variation in susceptibility to antimicrobial agents. Hence, it is necessary to determine the susceptibility of pathogenic bacteria isolated from the clinical specimens. Classification, disc diffusion method, that is Kirby-Bohr disc diffusion method and Strock disc diffusion method. Second one is a dilution test, that is Broad dilution method and agar dilution method. Third one is a E-test. Fourth is automated method and fifth is molecular method, PCR detecting resistant gene. Disc diffusion method, most widely used method, suitable for rapidly growing pathogenic bacteria, unsuitable for slow growing bacteria, and this method uses filter paper disc impregnated with appropriate concentration of antibiotic solution. Test bacterium is inoculated on the solid medium and then the discs are applied. The disc in the antibiotics in the disc diffuse through the solid medium. Concentration is highest near the site of the application of the antibiotic disc and decrease gradually away from it. Media, Muller-Hinton agar, best medium to use for routine susceptibility testing of non-fastidious bacteria for the following reasons. It shows acceptable batch-to-batch reproducibility for susceptibility testing. It supports satisfactory growth of most non-fastidious pathogens. It has minimal inhibitory effect on sulfonamides and trimethoprim. Hence, these antibiotics are better tested in MHA than other media. Modifications of MHA. Lysed horse blood is added to MHA to support the growth of fastidious organism like H. influenzae. 4% NaCl should be added to medium for testing MRSA isolates. Inoculum preparation. Isolated pure colonies of the tested organism are inoculated in suitable liquid medium, peptone water broth, incubated 35 to 37 degrees centigrade for 4 to 6 hours. Density is adjusted to turbidity with the 0.5% McFarlane opacity standard tube. Ideal inoculum after overnight incubation gives even semi-confluent growth. Too heavy inoculum reduces the size of inhibition zone. Lawn culture. Following ATCC, American type culture collection strains are used as a standard control strain. They are the E. coli ATCC 25922, Pseudomonas aeruginosa ATCC 27853, Staphylococcus aureus ATCC 25923, Enterococcus faecalis ATCC 29212. Antibiotic disc. It is a 6 mm diameter that is impregnated with standard quantity of antibiotic solution. Choice of it. Antibiotics should likely to be used for therapy of infections, local prescribing habits of the antimicrobial agents, resistant pattern of the locally prevalent pathogens, cost, toxicity, pharmacokinetics, and spectrum of activity of an antimicrobial agent. First line drug. Commonly used for the treatment, antibiotic currently being administered to the patients. Spectrum should be relevant to the organism isolated. Second line, that is restricted only to special circumstances. Reserved for testing later if the organism is found to be resistant to all the first line drugs. Kirby board diffusion method. Cotton swab is dipped in inoculum and squeezed to drain out the excess fluid. Swab is inoculated onto the MHA plate by lawn culture. After drying the surface of agar plate for 3 to 5 minutes, antibiotic discs are applied using either sterile forceps or multi dispensers. Plates are incubated at 37 degrees centigrade 16 to 18 hours. When tested for MRSA, results should be read after 24 hours of incubation. Zone size is measured using roll of vernier calipers that is shown in figure. Interpretation of zone size into sensitive, intermediate or resistance is based on the standard zone size interpretation chart. Commonly used disc concentration and interpretation of disc diffusion test. That is a diameter of zone inhibition in mm, resistant, intermediate and sensitive. Antimicrobial agents for benzyl penicillin, S. aureus, cefoxitin, gentamicin, amikacin, erythromycin, tetracycline, nitrofurantoin, ciprofloxacin, ceftriaxone, imipenem and vancomycin for enterococcus. Stokes disc diffusion method. MHA plate is divided into 3 parts. The test organism is inoculated onto the central 1/3 and the control strain on upper and lower third of the plate. An inoculated gap of 2 to 3 mm wide should separate the test and the control area on which the antibiotic discs are applied. Plates are then incubated at 37 degrees centigrade for 16 to 18 hours. Primary disc diffusion test performed when results are required urgently and single pathogenic bacterium is suspected in the specimen. Directly performed from a clinical specimen, result of the primary test should be subsequently verified. Now dilution test, MIC minimum inhibitory concentration, lowest concentration of antimicrobial agents that will inhibit the visible growth of microorganism after overnight incubations. That are the broth dilution method and agar dilution method. As in figure shows, test organism is inoculated in test containing serial dilution of an antibiotic, that is a 64, 32, 16, 8, 4, 2, 1 and control. Subculture is done on solid media by these test tubes No growth in first 2, growth is present in second 2. MIC is 8 microgram per ml, MBC is 32 microgram per ml. Serial dilution of the antimicrobial agent in MH growth. Each tube is inoculated with a fixed amount of suspension of test organism along with controls. MIC is determined by noting the lowest concentration of the drug at which there is no visible growth. Minimum inhibitory bacterial concentration can be obtained by subculturing from the each tube onto a nutrient agar plate without any microbial agent. The tube containing the lowest concentration of the drug that fails to show growth on the subculture is the MBC of the drug of that test strain. Both dilution test can also be done using microtiter plates, microbroth dilution method. Clinical applications of MIC. For confirming the AST result obtained by this diffusion test. For testing microbial sensitivity, antimicrobial sensitivity of slow-growing bacteria such as tubercular bacilli, bacteria for which diffusion test is not standardized, when a very small degree of resistance has to be demonstrated, when the therapeutic dose of the drug has to be regulated accurately as in the treatment of bacterial endocarditis. Serial dilution of the drugs are prepared in molten agar and poured into the petri dish in agar dilution method. Test strain is spot inoculated. Advantage or both dilution is several strains can be tested at the same time by using the same plate. Directly measure the MBC. There is no need of subculturing as it is done with broad dilution method. Epsilometer E-test uses an absorbent strip containing predefined gradients. Serial dilution of antibiotic concentration immobilized drugs along its length applied to a lawn inoculum. Antibiotic concentration at which the ellipse edge intersects the strip is taken as a MIC value. In figure, MIC is 1.5 microgram per ml. Automated antimicrobial susceptibility test. Automated system are available by Vitek2 identification and antimicrobial sensitivity. That is a Biomurex Phoenix system backed on Diskinson. Microscan walk-away system. Principle is microbroth dilution. PCR-based assay are available targeting specific drug resistance genes. For example, MAC-A gene for MRSA detections.